At the outer end of each arm of the antibody molecule, a specific amino acid sequence exists. This is where the antibody molecule reacts with the antigenic determinants in epitope, as explained before, that provoked its production. The combining site is known as the fragment antibody binding region. The most common antibody molecules have two Fab regions and are said to be bivalent, having two combining sites. So this is the antibody molecule. Antibody are proteins that bind very tightly to their target's antigen. It's like a hand which holds the objects. And this is the heavy chain, and this is the green one, the light chain, and this is the Fab region, fragment antigen binding, which is like a hand which is hold an object, and this is called the hinge region, which is designed as a spring which is permit the movement of the antigen binding site on the effector site, constant region. Antibody has its own specificity. So this object, yellow one, is the antigen, which is different in its shape from this antigen, which is different from its shape from this antigen, which is different from its shape from this antigen. So the Fab region of the antibody must change its shape in order to fit the shape of the antigen, and this can be achieved by changing the amino acid sequence in the variable region of the antibody. So this can fit the shape of the antigen, and this is known as antibody specificity. And this is called the paratope, which is a small region of about 15 to 22 amino acids of the antibody's fragment variable region and contains parts of the antibody's heavy and light chains. And the affinity is the strength of the interaction between single epitope and single paratope. When it is complete similarity between the shape of the antigen and the antibody, which is mean epitope of the antigen and the paratope of the antibody. So it is called high affinity. So it is called high affinity, while when there is difference in the shape between the antigen and the antibody, so it is called low affinity. Second part of the antibody, which is the constant region, which is the stem of the Y shape. This is the stem of the Y shape of the antibody. Links the antibody to other participants in the immune defenses. This area is identical in all antibodies of the same class, and it's called the constant region because the amino acid sequence in it is similar and not change, and it represents the carboxy end of the polypeptide chain. This region of the antibody molecule is called the FC region, fragment constant, the abbreviation of a fragment constant, because it can be crystallized. It is amino acid content and sequence is relatively constant and characteristic of its class. This portion of the molecule activates the complement system and encourages the phagocytosis. According to constant region of the heavy chain, we have 5 classes of the antibodies. It's called the isotypes, which is IgG, IgM, IgA, IgE, and IgD. The constant region of the light chain is 2 types, which is either kappa or lambda. The antibody is a protein, so it is like any other proteins, is degraded by the enzymes, which is called proteolysis enzyme, degraded the antibody into different fragments. And first enzyme, the papain, that split antibody at hinge region above the interchain disulfide bonds into 3 fragments 2 Fab and 1 Fc. So this is the antibody, and this is the hinge region. So the papain split antibody above the hinge region. Don't forget A for the above. A for the above. So we get 3 fragments, 1 Fab, 2 Fab, and 1 Fc region. While the another enzyme, which is called the pepsin, which is cut below the interchain disulfide bond and hinge region into 2 fragments. This is wrong. This is below here. This is below the disulfide bond. So divided into 2 fragments. So it's called Fab2. This is Fab2 and 1 constant region. Function of the immunoglobulins or function of the antibodies: activation of complement, opsonization, antibody-dependent cell-mediated cytotoxicity by NK cell, neutralization of toxin, agglutination of the RBCs. The immunoglobulins or isotypes had many classes. The classes of the immunoglobulin can be divided into 5 different classes based on differences in the amino acid sequence in the constant region of the heavy chain. IgG for the gamma heavy chain, IgM for mu heavy chain, IgA for the alpha heavy chain, IgD for the delta heavy chain, IgE for the epsilon heavy chain. While the immunoglobulin subclasses, the classes of the immunoglobulins can be divided into subclasses based on the small differences in the amino acid sequence in the constant region of the heavy chain. All immunoglobulins within a subclass will have very similar heavy chain constant region amino acid sequences. So the IgG subclass, IgG1, 2, 3, 4. And in case of the IgA, IgA1 and IgA2. Immunoglobulin types. Immunoglobulins can also be classified by the type of light chain that they have. Light chain types are based on differences in the amino acid sequence in the constant region of the light chain. Kappa light chain and lambda light chain. Now we will discuss each class, each isotype. IgG, the structure, this is the structure of the IgG, are made of 2 identical heavy chain and 2 identical light chain. It's monomer, only 1 antibody. Molecular weight about 150,000 Dalton. Called so because of its gamma heavy chain. The subclasses 1, 2, 3, 4 differ in the number of the disulfide bonds and length of the hinge region. Properties. IgG is the major immunoglobulin in the serum, constitutes 75% of the serum immunoglobulin. IgG is the only class of the IgG that crosses the placenta. IgG2 does not cross well, and this is of benefit during immunization of pregnant women. So the antibody will cross the placenta and will immunize the fetus, and the IgG fix the complement and mediate ADCC by NK cell. IgG4 do not fix the complement, binding to cell-like macrophage polymorphonuclear cells by Fc region of this is the Fc region of the immunoglobulin, and the antibody has prepared the antigen for eating by the phagocytic cells, which is called the opsonin. Main immunoglobulin in the secondary